Good morning students. Last class we had seen about the mitotic cell division. Today we are going to see about the meiotic cell division. So meiosis means reduction division. It was coined by first farmer in 1905. Then meiosis means the reduction means the chromosomal number diploid is reduced into half of the number, that is the haploid number. It takes place only in the sex cells or gametes. Understand? So it consists of two types, heterotypic division and homotypic division. Heterotypic division is nothing but the meiosis one, because the offsprings, that is the cells, the haploid cells are produced from the gametes or gonads is not resemble with the parent cell. So that is why it is called as heterotypic division. Understand? So the daughter cells are entirely different from the parent cell. So that's why we are called as heterotypic division. It divides into five stages. Prophase 1, metaphase 1, anaphase 1, telophase 1, and finally dikinesis. Understand? The prophase 1 is furtherly divided into leptotin, zygotin, pachytin, diplotin, and dikinesis. Understand? So it is the longest process of the meiosis. The prophase 1 is a longer duration. Leptotin, this is the first stage. The chromosome become uncoiled and assumed as the thread-like structures. Thread-like nool mare erko. Understand? Thread-like structures and take up a specific orientation inside the nucleus. It is known as bucket stage. Then next one is zygotin. Zygote means adjoining. That means adjoining. Understand? The homologous chromosomes approach each other and begin to pairing of the homologous chromosome, that is called as synapses. What is the meaning of homologous chromosome? Homologous means same chromosome. The structure and the functions are same, that is called as the homologous chromosome. So pairing of homologous chromosome takes place during the zygotin stage, that is called as the synapses. Next stage is the pachytin. So pachytin means the chromosome will be thicker, that is called as pachytin stage. Understand? So the chromosomes are as long as and visible in size. The pairing of chromosome takes place. The pair means two. That is called as bivalent. This is one bivalent, this is another bivalent. Bi refers to two, or pairing of chromosome takes place. Each bivalent contains tetrads, that is 4 chromatids. This is called as chromatids, this is called as centromere. So 4 chromatids, so that is why called as tetrads. Understand? Tetrads. Then homologous chromosome begins to separate. That is the chiasmata. That is formed the X-shaped. Interchanging of chromatids takes place. This is called as chiasmata. That is interchanging of the chromatin material. That is nothing but the genes are mingled with each other. That is paternal chromosome as well as the maternal chromosome. What is the meaning of paternal chromosome? The sex cells coming from the father, that is called as paternal chromosome. The sex cell, nothing but the egg from mother, is known as the maternal chromosomes. That will be a mixing of these two. We are called as chiasmata, that is otherwise genetic recombination takes place. There will be mixing of paternal and maternal chromosomes takes place in the pachytin stage. Then next one is diplotin stage. The chromosomes are separated. That is the homologous chromosomes repel each other, and we are called as terminalization. That is pre. So that is takes from any one of its point, either midpoint or end point, whatever will be, that is called as the terminalization. Then the chromosome from centromere towards the end. We are called as the terminalization. Then dikinesis. The paired chromosomes are shortened and thickened. The nuclear membrane nucleus begin to disappear. Spindle fibers make their appearance. So these are the stages we have met in prophase 1. Understand? Then the diagonesis is followed by the next stage, metaphase 1. The chromosome towards the equator region. This is the equator region. Understand? And finally, they orient themselves on the equator on the satire. The two chromatids do not separate. Two chromatids does not separate from the centromere. Spindle fibers make their appearance. Spindle fibers on thirpi aranchu. Then next one is anaphase 1. Each homologous chromosome with its two chromatids and undivided centromere move towards the poles. This is the north pole, this is the south poles. Understand? Move towards the poles of the cell. This stage in the movement is called as a dyad. Then telophase. The haploid number of chromosomes after reaching their poles become uncoiled and elongated, and the nucleolus reappear. This is the nucleolus reappear. Understand? And two daughter nuclei are formed. Then cytokinesis, the division of cytoplasm. Division of cytoplasm we are called as cytokinesis. The cytokinesis always takes from right angles to the left. Then the resultant is two daughter cells are formed. These two daughter cells are not resemble their parent cell. So that is why we are called as heterotypic division. So still now we have seen about the meiosis 1. So at the end, two daughter cells are formed. Understand? Next one is meiosis 2. The two daughter cells formed during meiosis 1 is further divided into 4 cells. The first stage is the prophase 2. That is, the meiosis 2 is always known as mitotic division. What is the meaning of mitotic division? Mitotic division means multiplication division. The two daughter cells are formed into 4 cells, divided into 4 cells. So that is why we are called as mitotic division. It is also called as homotypic division. Homotypic division means the offsprings, the daughter cells are as like as its parent cell. So that is why we are called meiosis is the homotypic division. Understand? Then the first stage is the prophase 2. The centrioles divides into 2 and moves to opposite poles, and also the astral rays are appear. Nucleus membrane and nucleolus disappear. Understand? This is the prophase 2. Then which is followed by metaphase 2. The chromosomes get arranged in the equator region and move to opposite poles. The two chromatids are separated. And anaphase 2, these are moved towards to the chromosome separated into daughter chromosome, and these are called daughter chromosomes, and moves towards the opposite poles. Then finally, that is telophase. The daughter chromosomes are centered. Chromosomes in the central vandirudhu. The nuclear membrane and nucleoli appear. This is the telophase. And also cytokinesis. The division of cytoplasm is known as cytokinesis. The cytokinesis divides into 2. At the end of this, 4 haploid cells are formed. Why? During meiosis, haploid cells are formed. Understand? So due to maintain the chromosomal number. For example, each and every organism have a constant number of chromosomes. For example, you are having 40 4 plus XX and XY, that is 46 or 23 pairs. To maintain the number of chromosomes, the gametes are formed into haploid set of cells. What is the significance of meiosis? To maintain the chromosome number, and also variations takes place. Variation takes place because the meiosis one is the heterotypic division. Understand? What are the differences between the mitosis and meiosis? The mitosis takes place in the somatic cells only, means body cells. Understand? But the meiosis takes place gametes or sex cells, gonad cells takes place. The mitosis takes place and the resultant the cell is also having diploid in condition, but in meiosis haploid conditions, that is 2 number of chromosomes, only 1 number of chromosomes. The mitosic cell division result the parents, the daughter cells resembles the parent, but the meiotic division, the daughter cells does not resemble their parents. Due to mitosis, after the mitosis over, growth takes place, only the gamete formation. After meiosis, gamete formation takes place. Mitotic division, only 1 division takes place. Here 2 division, meiosis 1 and meiosis 2 takes place. Understand? And identical daughter cells are formed in mitotic. Here the daughter cells are non-identical. Thank you, students. With this, the lesson is over.